Good afternoon. I'm excited to share the role of INPP5D in late onset Alzheimer's disease. I don't think I need to go into a long background of what Alzheimer's disease is, but we do know that it consists of amyloid beta deposition in the form of neuritic plaques, as well as neurofibrillary tangles, which are hyperphosphorylated protein tau, followed by neuronal loss and atrophy prior to any of the cognitive symptoms being present. What we also know is that there's microglial activation that occurs sometime after the deposition of amyloid beta, or at least that's the hypothesis. So a lot of folks, including our lab, have really focused on the microglial activation and microgliosis in this particular pathway. So what we know from large genome-wide association studies that there are several genes that put one at risk for developing late onset Alzheimer's disease. We know there are three particular genes shown up here, presenilin 1, presenilin 2, and APP that will cause Alzheimer's disease. There also are a whole host of other genes that are known to increase the risk of developing late onset Alzheimer's disease. And each of these are color-coded for a particular pathway which they are involved in. For instance, APP metabolism, tau metabolism, cholesterol, or in blue, the immune response. And what I think you can appreciate from here is that nearly 50%, just over 40% of these genes are associated with the immune response. And one in particular we've heard a lot about previously is TREM2. But we also highlighted on the right side another gene that's quite common in the population, which is INPP5D, which also is related to the immune system. So what is INPP5D? Without going into a long background in the cell biology of this particular target, INPP5D functions as a lipid phosphatase. It is selectively expressed on myeloid cells and serves as a regulator of immune response. INPP5D, also called SHIP1, is located downstream in this pathway that involves TREM2. So actually, TREM2 is activated, which results in clearance of some of its ligands, which would be cell debris, A beta, or other lipoproteins. So the function of INPP5D or SHIP1 is that it actually limits, it's inhibitory on the TREM2 cycle. So SHIP1 is an inhibitor of the TREM2 pathway. And INPP5D has been associated with Alzheimer's disease with an odds ratio of about 1.08. There's been some studies in the Japanese population of Alzheimer's disease subjects showing that INPP5D expression results in cognitive decline. So increased expression results in cognitive decline in Japanese AD subjects. And furthermore, there's been additional studies here at IU that have shown that INPP5D is associated with cerebral blood flow. So we have many reasons to think about what the role of INPP5D may be in Alzheimer's disease pathology. And then furthermore, is there a role or a potential target of INPP5D in treatment for Alzheimer's disease? So before we could really get into any animal studies, we went back to the human databases and looked at INPP5D expression. So we utilized tools in the AD Knowledge Portal, specifically the Accelerating Medicines Partnership for Alzheimer's Disease, or AMP-AD, has collected data from the Mayo Clinic, Mount Sinai, and the Religious Order Study and Memory and Aging Project, or ROS-MAP. So with the help of Kwanzek No and Andy Tsai, when he was here, we started looking at the expression of INPP5D in multiple brain areas in the late onset Alzheimer's disease brain. And what we found that regardless of Alzheimer's disease in multiple brain areas, we see there's a significant increase in INPP5D expression that's associated with an Alzheimer's disease diagnosis. And furthermore, that increase in INPP5D expression, which is on the y-axis, positively correlated in multiple brain areas with A beta plaque density. So we started thinking about this particular pathway, and Peter Lin in the lab is a med neuro student who's completing his dissertation on this work. Started thinking about the INPP5D pathway and its role in the TREM2 activation of the microglial response to amyloid beta. And INPP5D, the function of INPP5D has a lot to do with the downstream signaling pathways, where if it's inhibiting this pathway from proceeding, it can really hinder cell survival or cell morphology migration, phagocytosis, proliferation, and differentiation. So Peter looked at this target, and we know that INPP5D in Alzheimer's disease, at least late onset Alzheimer's disease, is upregulated, which we hypothesize results in a decrease in the function of the TREM2 pathway, thereby resulting in increased plaque accumulation. So we think that INPP5D reduction may modulate this microglial signaling pathway, which then would regulate these disease-associated microglia in Alzheimer's disease, thereby attenuating the disease pathogenesis. So if we inhibit INPP5D, perhaps we can increase TREM2 function, which would activate these downstream signaling pathways, which could increase phagocytosis, thereby reducing the plaque accumulation. So the first step in this process was, do we actually have an animal model that we can utilize to study INPP5D expression in Alzheimer's disease? So we looked at what we have in the laboratory, and one of these models is the 5X-FAD, which is a rather aggressive animal model of amyloid pathogenesis. So it contains 3 mutations in APP and 2 in presenilin 1. These animals, as I mentioned, are quite aggressive with the pathology, beginning to develop amyloid around 2 months of age. And we can begin to observe deficits in cognition related to this deposition by 6 months of age. So if we look between 2 and 12 months in a 5X-FAD, which are in red, compared to the wild type control that are in blue, we start to see increases in INPP5D expression around 4 months, and they're consistently upregulated through 12 months in both the cortex here and the hippocampus. And this is also shown down here in the westerns, and where we see increases in INPP5D at 4 and 8 months, for instance, in these particular animals. So this is expression in the cortex. We also can look in the brains of these mice, and we've shown that INPP5D here in green, IBA1 in microglia, and 6E10 in the plaques, which are diffuse plaques, and X34 in the more cord plaques. We see clearly in the bottom images when we merge them that the INPP5D expression is highly localized to the plaques that are, to the microglia that are associated with the plaques. At least this is in the cortex, and we also see the same changes in the cebiculum. And we published this last year, if you're interested in reading the details of the study in more depth than what I'm able to go over today. The exciting part, I think, is when we were able to cross a mouse that's deficient in INPP5D to measure the amyloid pathogenesis. So we took that 5X-FAD model and crossed with an INPP5D heterozygote. We cannot use a total knockout of this model because it ends up having peripheral infiltration of B cells, but the animal is totally fine behaviorally, and the immune system looks normal in the periphery. So we are able to utilize the 50% knockout of INPP5D to produce the INPP5D hets on a 5X-FAD background. So the data I'm showing today is from 6 and 7.5-month-old animals. We did some cognitive testing, immunohistochemistry, and some bulk as well as spatial proteomics and transcriptomics. And I'm not able to get into a ton of detail on those studies today. So we look at behavioral readouts in a 5X-FAD model. One of the most reliable behavioral readout in these animals is spontaneous alternation, which describes the tendency to alternate between these arms in pursuit of a stimulus in consecutive trials. So it's really utilized to investigate exploratory behavior and cognitive function. So yes, this is spontaneous if it goes into this arm and then into this arm and then can go back. But if it goes from this arm to this arm and then back, that's failing spontaneous alternation. So if we look at 4 months, we actually don't see any changes in any of our genotypes. But by 6 months, and this is the time point the 5X FAD does have behavioral deficits, we see over here that in black bars, the total black or just a black 6J, the KOWT here is an INPP5D het. So we don't see any behavioral changes between the black 6 and the INPP5D heterozygote. Here, the bar I'm showing you here, wild type is on the 5X FAD background. So this is a 5X FAD with normal levels of INPP5D compared to here, the heterozygous knockout on the 5X FAD background. So here's the animals that are the 5X INPP5D heterozygous. And what I think you can appreciate is that an animal that has normal INPP5D on a 5X FAD background has significantly worse percent alternation, meaning cognitive deficits by 6 months of age. And if we reduce INPP5D by just 50%, we can recover the spontaneous alternation in such that these animals on a 5X FAD background at 6 months do not differ from an INPP5D het or a black 6J mouse. And we see this in both males and female mice. Interesting, the underlying pathology may be indicative of some of these changes. And we can look at just a 5X FAD on the left and the 5X INPP5D het on the right. We've stayed on the top for diffuse plaques, in the middle for cord plaques, dystrophic neurites here in LAMP1, and neurons here in channel 4. And then we've merged them all together at the end. And what I think you can appreciate just by looking at 6010 and the INPP5D het and 5X FAD, you can see that there's reductions in both the cortex and the hippocampus of the 5X FAD INPP5D hets. We see the same changes in the cord plaques as well as in dystrophic neurites. We do not see any change in the neuron numbers, suggesting that the INPP5D deficiency actually reduces diffuse and cord plaques as well as dystrophic neurites in these mice at 7.5 months. If we further look at the morphology and really about how these this particular gene may be changing the phenotype of these plaques, we can look on the top at the 5X FAD. Again, we're looking at diffuse and cord plaques here, as well as neurons. And on the bottom panel is those 5X INPP5D hets. We actually start to see a change in the ratio of diffuse plaques and cord plaques between the 5X FAD and the INPP5D het, such that we see more of these diffuse plaques and less diffuse plaques and more cord plaques in the INPP5D heterozygous animal. And you can see that quantified over here, suggesting that the microglia are actually functioning to compact that plaque and not increase the spread of amyloid beta as you see in a 5X FAD. I think you can appreciate this too if we look at primary mouse microglia and look at the phagocytosis assay. So if we expose the primary microglia to fibrillar A beta 1 to 42 and then measure the co-localization suggesting an uptake of the amyloid beta 1 to 42, we can see over here on the right that in the heterozygous knockout microglia, we actually are able to increase phagocytosis about 2.5 times over black 6J microglia. This becomes further apparent if we start looking at other states of the microglia. So IBA1 we're utilizing as our marker of activated microglia, and P2RY12 is our marker of homeostatic microglia, and then there's merged on either of these. What you can start to see with a higher resolution confocal imaging is that in 5X FAD on the top look very different from the microglia that are interacting with the plaque on the bottom. So the plaque is in blue, and you have IBA1 microglia here. They're much more closely more closely up on top of the primary microglia, or up on top of the plaque, as well as the same, and you have more of these homeostatic microglia in the middle, and then you can see the merge on the bottom. And in fact, you can see here, although there doesn't appear to be overall more coverage of IBA1 activated or P2RY12 positive homeostatic microglia, the engagement, so the overlap, the co-localization of the IBA1 plaques is increased in the cortex and the hippocampus of the heterozygous knockout 5X-FAD mouse, suggesting again that we're changing the way that the plaque is being interacted with the microglia. And we can further see functionally if we measure cytokines, pro-inflammatory and anti-inflammatory IL-10, we can see that in the INPP5D het, we're actually also reducing pro-inflammatory cytokines. So we see reductions in IFN gamma, IL-1 beta, and TNF alpha, but an increase in pro-inflammatory cytokines like IL-10. So this data suggests to us that INPP5D expression in protein is upregulated in human late-onset Alzheimer's disease. These 5X-FAD mice can actually be utilized to model the changes we observe in human postmortem tissue. We've reduced INPP5D expression in vivo mitigates plaque burden in our 5X-FAD mice and further protects against behavioral deficits that have been previously induced by the amyloid pathology. We also think that the INPP5D deficiency alters the engagement of microglia to the plaques and maintains a more homeostatic phenotype, resulting in a pro-inflammatory cytokine release that's reduced. And lastly, attenuation of amyloid pathogenesis likely is related to the increased motility, proliferation, and phagocytosis of microglia that we observed with a reduction in INPP5D. And we are continuing this work with the TREAT-AD Center. INPP5D is a target of that particular center. So we are looking at beginning some in vivo studies with INPP5D inhibitors to determine if we can alter the disease pathogenesis. And none of this work is possible without a substantial crew of folks to help. And I'd like to thank especially Bruce Lamb, Gary Landreth, Wansik No, and Andy Tsai for their guidance really in this project, and for Andy, who performed some of the early work with the human data working with Wansik. The graduate students in the lab, Peter, Sam, and Disha, each are taking on some different aspects of tau pathogenesis or amyloid that we haven't even presented today. And then Cindy, Chris, Bridget, and Abby for all of their help in the lab, and the LARC staff for the care of our animals. And of course, all of the funding that this work has couldn't go on without, as well as the IADRC and the Rec Corps, Dr. Lahiri, Dr. Sakin, and John Spence, who helped tremendously the preparation of this work. And with that, thank you all for listening.